Good morning, everybody. Today we are doing chapter 8, which is Mendelian genetics. So we're going to get started in just a couple moments.
Morning.
Good morning, Bella. How are you? Good, good. I forgot that you don't have a microphone. Okay, you're here. It is just about 9 o'clock now, so I'm going to go ahead and start add people as they show up. Chapter 8, genetics. Let's get started. There we go. Okay. Resize some things here, make it so I can see everything well. Okay, here we go. Slideshow from the beginning. Okay, chapter 8, Mendelian genetics. All right, this is actually kind of a fun one. I hope that everybody's watching this and participating enjoys it, because now we're moving on from cellular, internal cellular stuff to stuff that has to do with you and me. And so it's kind of fun, kind of interesting. So I hope you like it. Okay, so why is it called Mendelian genetics? Because it was developed by a guy named Gregor Mendel. He was a monk in Abbey hundreds of years ago, and he had time on his hands, and he noticed that certain plants passed on certain factors or traits, and he sat down, because the time he had on his hands, to figure it out, and he did. And it's still the basics of what he said back then we still use to help understand how these factors, which we now know to be genes, are passed on. So he was the first to analyze patterns of inheritance. So prior to that, people had done a little bit of crossbreeding. They knew that two different plants coming together could make things good, but he made a study of it, and he did experiments using the scientific method, which is the way you do it. He deduced the fundamental principles of genetics. So he didn't know anything about DNA. He didn't know anything about genes, but he called them factors. And he knew that if he crossbred these plants with each other, he could predict what was going to happen, and he did. He studied garden pea plants, and these are the same pea plants that you get peas that you would eat as a vegetable. Each pea, by the way, that you eat is a baby plant, a seed that can grow into a complete new pea plant. Okay, so how does it work? Well, let's take a look at the figure here after we do the notes. So what he did was what is called cross-fertilization. Now, what you should know is that many plants are able to fertilize themselves. If there's no other plant around, they still have to make seeds so they can fertilize themselves. So what he did here is, if you can see the purple plant, he cut off the male parts. Ouch. No, it didn't hurt the plant, but that prevented the pollen on the male from fertilizing its own female portion of the plant. So he used pollen from the white plant onto the purple plant, and that's called cross-fertilization, when you have one plant fertilize another, because self-fertilization is another option. All right, Mendel then crossed two different true breeding varieties, creating hybrids. Now, what does true breeding mean? It means that these plants aren't hiding any other genes than what you see. So, for example, the purple would be nothing hiding, and the white would be nothing hiding. Now, we know that the white can't hide anything, and I'll explain how we know in just a moment. He also created true breeding varieties of plants. That is, making sure that there were no hidden genes or factors hiding behind the dominant, in this case, purple color. He tracked several characteristics in pea plants from which he formulated several hypotheses. Now, the traits that he tracked were flower color, whether the seeds were wrinkled or smooth, the color of the seeds, the height of the plant, things like that. So let's go through the figure. Okay, so what he did was he removed the stamens, which are the male part, from the purple flower. Then he transferred pollen from the white male, the white flower, the white plant, onto the purple female part of this purple plant. So what that did was, all of the genes from the white plant went into the carpel and fertilized all these little ova to become seeds. So in each of these seeds, there's genes from the white plant and genes from the purple plant. He called them factors. Now, when they grew, the pollinated carpel matures into a pod, and then he planted them. What he noticed was, after he planted them, what he noticed is that the offspring, which we call F1, standing for filial generation 1, were all purple. So the question was, if he knew that he put factors of the white onto the purple, where did those white outwardly appearing things go? And what he figured out, and what we now know, is that there's dominant and recessive genes. So let's keep going here. Okay. Mendel developed 4 hypotheses from the monohybrid cross. Now, he called it monohybrid because hybrid means mixing 2 very different things. Like a car, I just bought a car that's a hybrid. It's a combination of electric and gas. So hybrid means mixing 2 very different things together. In this case, he mixed white with purple. Monohybrid meaning just one factor. In this case, he was looking only at color, not at any other things. If he was looking at color and height, that would be a dihybrid cross. All right, there's alternative forms of the genes that are now called alleles. So, for example, there's a gene for brown-eyed color and a gene for blue-eyed color, and each of those is an allele for eye color. And an allele means a different version of a gene for the same trait, like eye color. If for each characteristic, each organism has 2 genes. Now, why do we have 2? It's because we have a mother that gave us a gene for that and a father that gave us a gene for that. Okay, even if a person is raised with 2 moms or 2 dads, biologically, they had to have a biological father and a biological mother, each of which contributed a gene for every single trait in the body, with the exception of sex-linked traits that we're going to be talking about later on. Gametes, like sperm and egg, carry only 1 allele for each inherited characteristic. I can only give half of what I am genetically to my kid because there's another person that also has to give half of themselves to the children. Alleles can be dominant or recessive. This is the basic understanding. We're going to find other more complex ideas about dominance or recessiveness, but from now on, I want you to think of dominant as always showing, and recessive can hide. It can hide behind the dominant. The genotype is what's actually inside the genes of the organism. Now, that doesn't always show on the outside. It's the hidden characteristics. The actual alleles are actual genes inside an organism's DNA. Now, the way it looks on the outside is phenotype. Think of it as like a photograph. So the genes that I have inside my body may not always show on the outside. I may have some genes that are hidden. So the phenotype is how the organism expresses the genotype within their cells. If you remember, we saw something similar. It was called the karyotype. And that was looking at the actual chromosomes inside the cell. But this genotype and this phenotype has to do with whether you're looking at the actual genes or what the person or organism shows on the outside. Okay, so let's look at this monohybrid cross again. This is going into a little bit more detail of what he did in that first figure. Monohybrid cross is a cross between parent plants that differ in only one characteristic, in this case, the color of the flowers, purple and white. The characteristic in this cross is flower color and which color is dominant. So if you take genes from the white and genes from the purple and mix them together, and all of the F1, which stands for filial generation. Think of the word filly as a baby horse. So filial means offspring. So in the first generation of a cross between these two, the white hides because it's recessive. So the dominant always shows, and if there's a white gene, it would hide behind the purple. Now, if you breed these purple plants with each other, what you find is what many people have called skipping a generation. So the white skipped a generation because it was hiding behind this purple allele, or purple gene. But when you mix them together, some of the white genes find each other in the offspring, and there they pop up again. So the purple flowers, which are dominant, white flowers, which are recessive, then all the offspring have purple flowers because the white is hiding. If you cross these with each other, have this F1 crossed with each other, you'll find that 3/4 of the plants have purple flowers. I personally prefer 75% or 0.75. I'm not a big fan of fractions, but either way, we're talking about the same number, and 1/4 of the plants have white flowers. Now in lab, which I do expect you guys to come to, we will be doing these. They're kind of like math problems. I know it sounds daunting, but I'll be showing you how to do a genetics problem, and you'll understand why this is the case. Okay, so how exactly does that work? Let's take a closer look. All right, so if we look at the top of this, the genetic makeup or genotype is the alleles. Now, the question is, why do we use big P and little p if it's purple and white? Well, the idea is the letter indicates where on the chromosome we're talking about. So the allele for purple sits in the exact same spot as the allele for white would if it were a white gene. So we use the first letter of the dominant allele. In this case, the word purple starts with P. So instead of using P and W, we use capital P and lowercase p. Capital P for purple, lowercase p for white. And that's just the convention. Now notice that this P over here is underscored. Why is it underscored? Well, In typing, it's not that big a deal because it's very obvious whether you're looking at a uppercase or a lowercase. But when you're writing on the board or on a piece of paper, a capital P looks very similar to a lowercase p if you just write it right in the middle of the paper. So what I ask my students to do is for any letter where the capital and the lowercase have more or less the same shape, underscore the lowercase letter. For example, X is a good example of that. Or we have P, O is another one. T is different because T has the cross more in the middle for lowercase and on the top for the uppercase. So that's when you don't have to underscore. But when the capital letter and the lowercase look really, really similar, you underscore the lowercase only when writing it on paper. On a computer, it's obvious because the font size is standard. Okay, so let's take a look. This is a double dominant purple plant. So when it has to decide which of these genes to give its offspring, it doesn't have a choice. It's always going to give a big P. So we're going to get to that in a moment. Let's do the nose. A Punnett square is seen here. This is kind of more like a Punnett diamond. I like to do it as a square that's flat on top and the sides. A Punnett square is used to determine the possible genotypes and possible phenotypes of offspring possible during a cross between two organisms. So you're kind of like trying to guess what's going to happen if we cross these two organisms, in this case, plants. The P plants are the parents. Think of P for parents. So these are the first parents that we start with. This is the top row. F1 means offspring filial generation 1. So these are the first, the outcome of the first cross. F2 means offspring generation number 2. Usually it's a cross of the F1 generations as parents. So you crossbreed with the offspring. I know that sounds kind of gross. And we're going to talk about why incest is taboo and gross later on when we talk about human genes. Okay, so let's do some of these. Now we have 2 purple alleles or purple genes in this purple plant because there's nothing hiding there. So it can only give a capital P. But the white plant has 2 white alleles. Now, why is that the case? Now, this is really important. You guys pay attention. White can hide behind purple, but purple never hides. Dominant genes never, ever hide. If they're present, they always show. So I'm going to be repeating this with you in lab. It's really important you guys get this. And that is that dominant genes cannot hide. Now, up here, since there's 2 purple genes, it doesn't matter which one shows. But neither one is actually hiding because they're both being expressed. In this case, we know there's no purple gene because the purple would show and the flower would be purple. So with recessive genes, the only way to show it is to be double recessive because dominant doesn't hide. I hope you guys remember that. Try to make a special note of it because you're going to be tempted to think dominant can hide and it doesn't ever. So there you go. Okay, so now and what we're talking about, gametes here are sperm or egg. So remember that when you get half, all capital P, all dominant from the purple plant, all recessive lower P for the white allele. And when you put them together, all of the offspring are purple, but with a hiding white allele from the white parent. Now, these offspring, when they breed, they do have options. That means they can either give a dominant big P for purple to their offspring in the gametes, or they can give the little p to their offspring in one of their gametes. So if you have a mix, you can give one or the other, and about half of all the gametes end up with dominant and half end up with recessive. Now, when you put these together, now there's options. Now there's things that are different. So what we do is we take this genotype here and we split it up. We put the first letter, which is often dominant, in the first spot. So the big P goes right there. It's kind of like a times table. The little p goes down here. Now, I didn't underscore this, but the little p goes here. So this p goes there, this p goes there. Then we do the same for the other plant, which is also this. So we're crossing 2 of these with each other. So the big P goes first, the little p goes second. Now, one of the conventions with Punnett squares is we always put the genotype, which is the letters, on top, and then the way they look down below. In this case, they've put the way that it looks as a background to the box, but either way, you know what the offspring looks like because they've shown 3 of them are purple and 1 of them are white. What we're going to see is not all the purple are the same. So you do cross multiplication here. Big P and big P becomes 2 big P's. This is called homozygous dominant. Homo means the same. We're going to go into those definitions in a moment. But when we cross multiply here, the big P comes down to this box, little p goes here. This is still purple. Why? Because the dominant shows and the recessive hides. So notice these two look the same on the outside, but they're very different on the inside. In this case, the phenotype would be the same, but the genotype is different because what's on the inside is different. If we do the same cross multiplication here, same thing. But look what happens. These white ones come down, and now the white color has skipped this generation and shown up here because each of these have a chance, 1 in 4 chance, that they both will give the recessive. And so this recessive pops back out. And this is why in some families, you have two dominant brown-eyed people can have a blue-eyed child. Neither one cheated on their spouse because they were both hiding a blue-eyed gene. We're going to get to that in just a bit. So the phenotypic ratio. Now, what do we mean by a ratio? How many are dominant versus how many are recessive? So the ratio is always written as a number and then a colon and then another number, with the dominant being first and the recessive being second. So in this case, 3 purple, 2 TO, 1 white. So 3-to-1 ratio, 3 purple to 1 white. You can see that we're talking about the pictures here, the colors. The genotypic ratio is different. Now, why is it different? Because if we clump all these 3 here together, we get all these 3 purple ones. But notice, not all the purple are the same. So we have 1 double dominant purple, which is right there. We have 2 heterozygous, which means mixed, here and here. But they all look the same on the outside. And then 1 double recessive, which is called heterozygous, a homozygous recessive. And that one shows up as white. So we're going to be doing the details of this in lab, but here's your introduction. All right. Now, you guys may remember this figure from our previous chapter on meiosis. And what we're going to be talking about is how does the process of meiosis lead to some of these principles, the principles of segregation. Now, segregation means to separate. Now, in our history, in our country, segregation was not a good thing because we were trying to separate different races. But segregate just means to separate. And in this case, we're segregating or separating the genes that came from your mom from the genes that came from your dad. So pairs of alleles segregate or separate during gamete formation. Remember, they line up side by side. What you got from your mom is here, and what you got from your dad is here, and they always separate. So that means that 2 of mom's alleles for one trait, like eye color, can't go to the same kid. They always separate. So 1 from mom and 1 from dad come to you because mom gave 1. Dad gave 1. All right, moving on. Okay, homozygous and heterozygous. I alluded to this earlier. Homo just means the same. We use that for homosexual. We use it for homogenized milk. So the prefix homo can mean human, as in Homo sapiens, but it also just means the same. So in this case, homozygous means both traits in the zygote. Notice this word zygous comes from zygote, which is the cell that forms when the sperm and egg unite. So in the zygote, both are the same. So you can have 2 dominants or 2 recessives, but in either case, they're the same. So what we use for, now, unfortunately, blue and brown both start with the letter B, but the B stands for, the big B is for brown and the little b is for blue. In this case, it's the same, but because brown is dominant, the big B stands for brown, the little b stands for blue. Now notice these are both homozygous because they have the same. So this would be homozygous recessive. This would be homozygous dominant. Heterozygous is a different term, though. Hetero means different, as in heterosexual. In heterosexuality, you have 2 different genders hooking together. So hetero means different. So in this case, you have 1 dominant and 1 recessive. Now, you don't have to call this heterozygous dominant because there's always 1 dominant, always 1 recessive. So that's just repeating something that you should know. So you don't have to say heterozygous dominant. You just say heterozygous, 1 dominant, 1 recessive. There you go. 1 dominant, 1 recessive. So if we look at this brown-eyed person, how can they appear brown if they have a gene for blue eyes? If you remember, the recessive gene for blue can hide behind the dominant. So a person who's brown-eyed may be hiding a gene for blue eyes. And this was the case with my mom. I'll be talking a little bit more about my parents later. My mom was hiding a gene for blue eyes behind her brown eyes. I got it. My dad, being blue-eyed, gave me his blue-eyed gene. And we're going to be talking about how I was able to deduce that my mom was hiding a blue-eyed gene by the fact that I have blue eyes. Blue eyes, excuse me. All right. So when we look at actual chromosomes, so these are the chromosomes separating during meiosis. A locus means a location, a place where you would find a gene. So in this case, this is the locus for gene P, the locus for gene A, and the locus for gene B. So the genotype is when we put these two alleles together when it's inside the zygote. So in this case, we have a homozygous dominant. Here we have homozygous recessive because we're putting two recessive together. And here we have heterozygous. You do not need to say heterozygous dominant because it's always one of each. So the book says homozygous for the dominant allele. We usually simplify that to homozygous dominant. This is homozygous recessive, and this is heterozygous. Okay. Using a test cross. So this is what happened to my parents without them knowing. So this was my mom with brown eyes, and this was my dad with blue eyes. So what is a test cross? Well, when you look at a dominant individual, in this case, a purple plant, or my mom, there's no way to know just from looking at the plant whether it's hiding a white allele or another purple allele, but you can test it. So when you first look at a dominant individual, you don't know for certain what it's hiding, and if you guess, it will be wrong. So please make a note of that. If you just say, oh, I'm just going to guess it's purple, I'm going to guess it's white, that is wrong. Okay? So if you don't know, don't guess, leave it blank or put a question mark. Now, how do we know for certain that the white plant is double recessive? Could it be hiding a purple gene? What do you guys think? Could it be? I don't know how to get to the chat here. The chat, can I get chat here? Well, it's not letting me see chat. Anyway, so if you said it, good job. But the reason why you know this is double recessive is because the dominant purple never hides. It's something you've got to get in your head. Purple never hides. It always is shown. This could be hiding a recessive white, and it could be hiding a purple. This one cannot be hiding a purple because it would always show. You cannot be recessive. So that's how I know I don't have a gene for brown eyes, because I would have brown eyes. A person can be brown-eyed with a gene for blue eyes because the brown dominant covers the recessive blue. So I know that I'm homozygous recessive because if I had the dominant brown-eyed gene, I would have brown eyes. All right. So the test cross is trying to figure out, what is this dominant organism or person hiding? A test cross is a mating between an individual of unknown genotype, this dominant one, and a homozygous recessive, which you always know because recessive only shows if it's homozygous. If any of the offspring from this trait, from this cross are white, what does it tell you about the purple parent? So let's say, so we're looking at two possibilities. If this purple plant is double dominant, then that means all of its alleles will be big P, and all the alleles from the white parent will be little p, and that means all the offspring will be purple. But if this purple plant is hiding the recessive, that's what its genotype is, and it means that half of its alleles are going to be big P and half are going to be little p white. And in this case, you have a 1-to-1 ratio, or half purple and half white. So if you have enough offspring and half come out white and half come out purple, that means you know it was hiding the recessive. If all of them come out purple, you can be pretty sure it was hiding another dominant. So that's what happened with my parents. My dad had blue eyes, he was double recessive. My mom had brown eyes, unknown. My brother was born brown eyes because he inherited my mom's brown-eyed trait, and he showed it. My brother's hiding a gene for blue eyes, and I know that because my dad has blue eyes, and he can't give a gene he doesn't own. My dad passed, but he had blue eyes. So I know my brother's hiding it, and I also know because some of his kids are blue-eyed. But anyway, so he's hiding that trait. My mom gave me her hidden blue-eyed trait, and my dad gave me his only blue-eyed trait, and that's why I have blue eyes. So that's how I was able to know that my mom was hiding that gene for blue eyes, because I got two, one for my mom, one for my dad. Here's another thing you got to get out of your head. It's not possible for me to get two from my dad and none from my mom. It doesn't work that way in genetics. It could happen in a very rare circumstance, but as far as your understanding of how genetics works, don't be tempted. It's always one from mom and one from dad. So if I got two blue, that means I got one from my mom and one from my dad, right? And that means if I got one from my mom, she had to have one. She shows brown, she was hiding a blue-eyed gene. We'll get more into this when we go into lab. Okay, so the rule of multiplication. The probability of a compound event is the product of the separate probabilities of the individual events. What does that mean? Well, if you guys know what the probability of getting heads on a coin flip is, 50% or 1/2. The odds of getting 2 heads is not 1/2. It's 1/2 times 1/2. And if you remember your basic math, that is 1/4. And why is that the case? Because each event has a possibility of deviating from what you expect. So there's a possible deviation on the silver coin and a possible deviation on this gold coin. So the only way to get 2 heads is if they both go the direction that you want. So that's out of 4 different kinds of flips. Only 1 out of 4 is going to be double heads. 2 out of 4 are going to be one of each because the heads could show up on either one of the coins. But still, you have a 1 head, 1 tail combination, heterozygous. The only way to get double tails or double recessive is to get this. So the odds are 1 in 4 for this, and 1/2 for 1 head and 1 tail, but 1/4 for the gold head and the silver tail, 1/4 for the silver head and the gold tail. And that's why locks need lots of possibilities, because, for example, a lock that has 0 through 9, if you only had 1 spot, you could guess it just by flicking 1 number and then pulling and flicking and pulling, and eventually you could get it. But every time you add another digit to this lock, the chances of randomly spinning it and getting the right combination go up exponentially, because if you have 2 digits, 0 through 9, it's 99 possible combinations. 3 digits, it's 1,000 different combinations. 4 digits, 10,000 combinations. So that's why passwords are supposed to have lots of numbers and letters, because the chances of guessing it randomly are very low, because there's all these spots where they can change and be different. Okay, so how do you figure it out? You multiply the probability of one thing happening with the probability of the other thing happening at the same time. So 50% and 50% equals 25%, because there's a deviation possible by each event, each coin flip. I'll show you how it works. We do this in lab coming up pretty soon. Okay, so these are human traits. Now, these are not diseases. These are just things that you should know. Mendel's principles apply to the inheritance of many human traits. Now, without even seeing you, I can tell how many of these traits you have. And if you think, well, how would Mr. Sage know without even looking at me? Well, the answer is you have 3 of these. And how do I know for sure? Well, look at them. You either have freckles or you don't. That's 1. You either have a widow's peak or a straight hairline. That's 2. And you either have free earlobe or attached earlobe. That makes 3. So you have 3 of these in whatever combination you happen to have. Okay? So the question is, do you have any of these traits? Yes, we all have 3 of them, one or the other. We either have freckles or don't, widow's peak or straight, free or attached earlobes. Do they affect your ability to marry, have children, and pass on your traits? No. Some people find freckles cute. Most people who dislike freckles are the ones who possess them. But a lot of people find them extremely cute. Widow's peak, no one even really cares and notices, and free earlobes, most people don't care or notice anyway. All right, we're going to talk about family pedigrees here. Family pedigree has nothing to do with dog food, by the way, so don't be tempted on the test to think it has something to do with dog food. They use that term pedigree in dog food because dogs have a pedigree, and you can check their parents and grandparents, etc., if you want a purebred. By the way, purebreds are always less healthy than mutts, and I'll explain how that works later on. Okay, what does it do? It shows the history of a trait in a family. Now, in this case, we're talking about congenital deafness. That is not deafness due to something that happens to you in your life, but something that you're born with. So what these pedigrees do is they allow researchers to analyze human traits. Now, what we're talking about here is the gene for deafness, which is recessive. Now, I want you guys to go through this with me carefully and slowly and take notes. Now, what we're looking at here are the grandparents, the parents, and the grandchildren. Dominant is shown by a light or white symbol, an empty symbol, usually in this case white, because you were writing on white paper. And then the darkened shape indicates the person who is showing the recessive, which means that they have 2 of them. The only way to show recessive is to have 2. You guys remember that? Got to have 2. So what we do is, unfortunately, they're using D for deafness, which is I don't like. And we're not going to do that in lab. But in this case, they're saying big D is hearing, little d is deaf. Females are shown as a circle, males as a square. So this is the convention on a pedigree, a human pedigree. Okay, so let's start with Joshua and Abigail. These are the grandparents. The question is, if they're showing the dominant, how do we know for sure they're hiding the recessive? Well, this is how you do it. And take notes on this. Look at Jonathan here. Jonathan Lambert are the children of Joshua and Abigail. He got 2 recessives, which made him deaf. Where did he get those from? Well, he had to get 1 from each parent, because you remember that's the rule. You always get 1 gene from one parent and 1 gene from the other. So he had to get this little d from one of the parents and the other little d from the other, and it doesn't matter which. But what that means is Joshua and Abigail had to be hiding those 2 for Jonathan to be born, because genes don't just magically crop up. That's not how science works. It happens because Joshua and Abigail were hiding it. Now, the question I have for you is, let's look at his sister, Abigail. How do we know for certain, why don't we know for certain what she's hiding? Most students would be tempted to write a little d because it kind of matches the parents. But here's my question. Is it possible that Abigail got a big D from Joshua and a big deal from Abigail, her mother? Yes. Is it possible that she got 1 D from Joshua and a little d from Abigail? Yes. We don't know. Since there's no recessive double recessive above her, and she doesn't have any double recessive kids, we don't know for sure. And remember, guessing in genetics is wrong. It will always be counted against you, so don't guess. If I say, why did you put a little d then? You go, I don't know. Then it's wrong. Don't do it if you can't justify it. Okay, but we did fill in Joshua and Abigail because we know that Jonathan got 1 of each of his from each of his parents. Same thing up here with John and Hephzibah. How about that for a name? Hephzibah, old school. All right. Now, even though Elizabeth, and I'll show you how we know Elizabeth has this, even though Elizabeth has 1 of each, we don't know which parent she got it from, so we don't guess. It's possible that John had 2 big D's and the mom had 1 recessive, and it's also possible the reverse. So we know Elizabeth got her little d from 1 of them, but we don't know which one, and we can't guess because that is wrong. So we leave these blank because neither one of these has a double recessive child, which would indicate they're both hiding it. Since Joshua and Abigail have a double recessive kid, we know they both had to hide it because he got it from 1 from each. Now, let's look at Elizabeth. How do we know Elizabeth is hiding it if we don't know what the parents are hiding? Well, look at what happened when she had kids with Jonathan. Now, Jonathan is always going to give his little d because that's all he's got. But she was able to give half of her kids a big D and half of her kids the other thing, which we now know is little d. How do we know? Well, look at this boy and this girl. They're both double recessive, both deaf. Where did they get their little d from? Well, they got 1 from dad, 1 from mom. Now, remember, dad can't give 2 and mom none. So this little girl and this little boy got 1 from dad and 1 from mom. That's how we know Elizabeth has a little d, because she had to give it to this boy and this girl. Who did she get it from for her parents? We don't know, so we don't guess. We know she's hiding it because 2 of her kids are showing it, and it's got to come from both parents. That's how pedigrees work. We're going to see it a little bit more in lab when you guys get here. All right, recessive disorders. For example, deafness. Most human genetic disorders are recessive. Now, why is this the case? Well, let me ask you a question. If you saw somebody that you were thinking about dating and there was something weird about them or something off, would that be attractive or unattractive if their eyes were not level, or if there was something about them that you could tell was off, that just didn't seem to work right? Well, most people wouldn't want to admit this out loud, but we almost all are seeking mates that have the best possible combination of traits. We want them to be smart and attractive and funny and able to hold a job and things like that, pretty much, right? That's what we want from our spouses. And if you see something in someone that's really obviously off, it might make them less attractive. And over time, if that happens over and over again, the chances that this issue that the person has preventing them from marrying and having children goes up exponentially, especially if it's a trait that kills you before you're able to have kids, because you can't pass on any trait to your kids if you die before the age of being able to have kids. So most recessive disorders, most human genetic disorders are recessive, especially those that are fatal, and especially those that are fatal before you can have kids. But individuals can be carriers of this disease. And I know how many of us are carrying a recessive disorder, and that's everyone. Everybody on this planet has at least one recessive disorder that they're hiding. How do I know for certain? Because there's lots of them and you can't tell. And you can't even tell if your parents were normal because they could have been hiding it. The only way to know if you're hiding it is if you have a kid that shows it, which means that you mated with someone who has the disorder or they were also hiding it. And I'll show you how that works. How did deafness pop up in this family, just like we saw Joshua and Abigail? Well, we have a normal person that doesn't know they're hiding a gene for deafness. This is the mom. She puts some of her eggs as capital, some of her eggs as lowercase, the recessive deafness. Same with dad. These are the sperm cells. He can give big D or little d. Now what happens is 3 out of the 4 kids are normal because they have the dominant gene for hearing, being able to hear normally. But in this case, 1 out of 4 times, if they have enough kids, there's likely to get one that's going to be deaf because they're both hiding it. And this is how you knew then mom was hiding it and dad was hiding, because they can hear just fine. But now this is the reason why we don't marry our siblings, our parents, or our cousins. Why? Well, when I had children with my ex, she was not related to me. Now, what does that mean? What it means is the hidden recessive, nasty problems that she had are different than mine because we're not related to each other. So the chances of something like this, deafness, popping up in our kids are low. But what are the chances that your brother or sister is hiding the same recessive as you? Very high, because you have the same parents. It's quite high even in your cousins because you have the same grandparents. So what that means is if you marry and have kids with your siblings, parents, offspring, or cousins, there's a chance that something nasty is going to pop up. And this is where the taboo comes in. Now we know, incest is bad, but why? There's got to be a reason. Well, it's just icky. Why is it icky? It's because our ancestors knew that if you mated with someone really genetically close to you in your family, the chance of some really bad thing happening in the offspring were pretty high. So it became taboo. That means we don't do it. We don't even consider doing it, right? Because it's going to be a problem genetically. So there you go. All right. So these are some disorders in human. Now notice most of the disorders here are recessive. Albinism, that is people who can't produce any pigment. Cystic fibrosis, galactosemia, PKU, sickle cell. Now we're going to be talking later on about why the gene for sickle cell isn't always bad, but that's later. And Tay-Sachs disease. Okay, dominant disorders. Now, this is kind of an issue because is dwarfism really a disorder or is it just a difference? So that's kind of a interpretation. If you guys ever seen the show Big World, Little People, or Little People, Big World, what it shows you is that dwarfs or little people can have very normal lives. So I don't necessarily think it's a disorder, but it is different from average or normal. So the authors of this particular text listed as a disorder. But look at this. Look at these Alzheimer's and Huntington's. How is it that if Alzheimer's kills you, it can be a dominant trait? Well, you got to look at where it says right here. Usually strikes late in life. What have most people done by the time they are 60 or 70 or 80? They've had kids. So I might be hiding a gene I'm probably hiding a gene for Alzheimer's myself, but I was able to successfully find a mate and have kids and pass it on without even knowing for sure, because it didn't affect my ability to be attractive to a mate. So the things that are deadly and dominant hit you after you have kids, because if it's deadly and dominant and hits you as a child, you die before you can have kids, and that gene dies with you. Does that make sense? So the only possible deadly dominant disorders are the ones that hit you after you reproduce, because then you've given it to your kids without meaning to or knowing it. Okay? There we go. All right, incomplete dominance is another topic that we're going to be talking about. You know what I think I'm going to do is I think I'm going to pause here. Give me just a moment, folks, and what am I doing here? How do I stop? How do I stop sharing? What am I doing wrong here? Here we go. Stop share. Okay. I think, let me just check something really quick. I think that we're going to be able to take this up. This is Bio 2. Let me just check something really quickly. Module 10. Yes, okay. So we are going to be finishing this chapter after the break. So what I want you guys to do is work on any review questions that you're able to finish based on what we talked about. So what we'll be doing in lab is we'll be starting genetics problems, doing the basic ones, and then doing some other ones as well. And then after we've covered those, we'll be finishing chapter 8, genetics, after the break. Today's our last lecture before the fall break. So I'll be seeing you back here 2 weeks from today. And I hope to see you guys on Monday after the break. After the break, remember, we don't have any classes at all. Nothing online, no lectures. We're all taking the time off. And so I'll see you back on Monday for the lab, and then Wednesday for a continuation of this lecture. Junice or Bella, do you have any questions about what I just covered? It was a lot, I realize. Okay, you guys good? All right, well, I do appreciate you coming. I hope it was informational and helpful and interesting on some level. I hope you guys have a great break. Work on your review questions. I hope to see you in lab on Monday. Have a good one, guys. Bye.